I'm going to spend some time giving you a short review of coronavirus cell entry, and then I will present some unpublished findings from our lab. We're back to molecular and cell biology now, some reductionist virology here, with a focus, of course, on coronavirus spike protein-directed membrane fusion. So that's the topic for the next few minutes. And so to start out, as depicted here, the biogenesis of coronavirus membrane fusion machinery begins at the virus assembly and egress stage, which is shown schematically at the far left. And that's the case because the viral spike proteins that operate in virus entry, they originate in the endoplasmic reticulum of virus-producing cells that are within the airways of an infected individual that's depicted at the top left. And these spikes accumulate with two other coronavirus membrane proteins, the M and the E matrix and envelope proteins, and then they assemble with a viral ribonucleoprotein complex into an intermediate exocytic compartment, the ER-Golgi intermediate compartment, and the budded viruses then secrete through a lysosomal egress pathway. High resolution EM images, which are cryo-EM, at the top right, reveal the spike proteins that are so fundamental to entry. This image that I put here is from a science paper by Turanova et al. And they made several memorable points. They and many others made several memorable points. One seen on these EM images is that the spikes aren't evenly spaced. There's not a dense forest of projections off these coronavirus particles. And another point within the paper and several others is that the spikes are highly dynamic structures with several flexible joints up and down the trimeric architecture. In fact, flexibility is referred by analogy to macroscopic joints, ankle, knee, hip. The key is that spikes can adopt several different configurations, something that's been known for some time, of course, but is actually an impediment to mass producing engineered stabilized spikes for vaccination purposes. So the flexibility is introduced into spike proteins during their biogenesis, most notably here in the Golgi, where proteases cut the spikes about in half, forming a peripheral called S1 fragment, and then a transmembrane, virion membrane incorporated S2 fragment. This is called the priming cleavage. Now, this is execution of the membrane fusion process. It begins with diffusion of viruses to susceptible host cell surfaces and virus binding then to attachment factors depicted in red here. And the notion is that viruses now restricted to two-dimensional surfaces can then diffuse laterally until they encounter protein receptors, ACE2 for SARS-CoV-2. The receptor binding domains, at least the ACE2 receptor binding domains in green here, are themselves highly dynamic up at the apex of the trimer. And so when bound to ACE2 receptors, they're restricted to a particular so-called standing conformation. And this standing conformation is thought to be unstable and may well facilitate then the wholesale release of S1 from S2. It's termed S1 shedding, depicted here with my arrow. And there's some evidence that S1 shedding can unleash the transmembrane fragments for unfolding into an extended so-called fusion intermediate state, which is depicted here towards the right, which probably holds then viruses to cells independent of the receptors. And at some stage, this extended intermediate subjected to a second proteolytic attack, the so-called activating protease cleavage, at a position that's a little bit more carboxy terminal to the priming cleavage, and that leaves a somewhat smaller transmembrane fragment that's able to then fold back upon itself into a hairpin form, in turn bridging the opposing membranes
close together, enough to fuse, and then genomes enter cells and infection ensues. So there's two, proteases are the triggers for fusion activation, not acid pH, which would be the case for many, many other enveloped viruses. So in further dissecting these steps, it's reasonable to start at the beginning with the initial attachment step, now shown at the far left. And here, sorry, go back one. Here it's kind of remarkable that there are so many attachment factors identified thus far for SARS-CoV-2. I mean, the spikes on coronaviruses are indeed multi-domain structures, so there could be several diverse binding potentials. So possibly this fairly long list of attachment factors is a reasonable list. I'm not sure I even have all the putative attachment factors listed here. It's kind of hard to keep up with the literature. There's so much, but here's some, the lectin sign proteins, transferrin receptor, scavenger receptor, TIM1, CD147, neuropilin-1, also sugars may be attachment factors, heparan sulfate, sialic acid. As I see it at this time, the field's in the process of sorting out which of these attachment factors, largely identified in vitro, are really the most meaningful to infection, particularly in vivo infection. And that's work that I would claim still needs to be done to sort out these attachment factors. But there's still many questions, and one recent PhD graduate student in our lab, Enya Ching, he spent some time addressing two questions, not specifically what the attachment factors are for SARS-CoV-2, but rather which viral spike protein domains bind to attachment factors. As I said, the spike's a multi-domain protein, so which domains bind. And then a question that's been on our minds for a while, does the initial binding to attachment factors, say, ready spikes or prime virus spikes in ways that make them more prepared to execute the subsequent stages of the fusion and entry process? And so I'll take a few slides. I'll describe his results today. All these results were obtained in the last few months. Enya's been working hard on these questions. So here's an entree to his data. In this entree slide, it makes kind of an obvious point, yet it sometimes seems to get overlooked in the lab, in our lab at least. These attachment factors, they're revealed in virus entry assays really only when target cells have low levels of protein receptor. In the case of SARS-CoV-2, low ACE2 levels. High ACE2 levels on target cells, as you would expect, they obviate the need for the often lower affinity pre-attachment factors, and the virus just binds straight to ACE2 and the rest of the entry process ensues. So Enya uses cells that either have very low, almost undetectable ACE2 levels or very high. And at low ACE2 levels, infection by viruses and pseudoviruses is also very low, sometimes below our assay sensitivity levels. So what Enya often employs are what we call cell-cell fusion assays, as they give readouts for spike-mediated fusion even when ACE2 is nearly absent. And his assay format is at the left here. He overlays spike-bearing cells onto target cells, oftentimes in the presence of interfering reagents, such as free soluble RBD receptor binding domains, FC RBD domains, or FC N-terminal NTD domains. These are components, of course, of the viral spike. If the spike and the attachment factor bearing cells upon contact really do fuse together, even, for example, in the absence of these interfering agents, then if they fuse together, then reporter fragments can come together. He loads up a fragment of luciferase in the spike bearing cells and a sister fragment in the target cells. And so if fusion ensues, reporter fragments can come together
and they complement to make luciferase, which is very easily measured. So at the right, you can see that Enya finds in these assays for spike-mediated cell-cell fusion, R-Luc luciferase as a readout. He finds that NTDs and RBDs as free soluble proteins both suppress some cell fusion when ACE2 levels are low. But when the ACE2 levels are high on the target cells, then really only the RBDs, which bind to ACE2 itself, will suppress. So we think this means that the NTDs can interfere with spike binding to attachment factors, albeit somewhat weakly in this assay format, but we take this as some evidence that the NTDs harbor virus, a site for low affinity attachment to cells. So the next question Enya went on to ask is, which NTD subregions are really necessary then for suppression of the fusion signals? And so at the right here is the spike ectodomain structure as resolved, of course, in several labs. The structural biologists have, of course, made enormous contributions to COVID-19 research, and David Wiesler, Jason McClellan, Andrew Ward, several others have used cryo-EM to get these high-resolution structures. And so note the green RBDs at the virion distal regions of the trimer, and the NTDs at the outer regions of the S1 caps, and in gray is at least a portion of the stalk-like S2. What's notable about SARS-2 is that the NTDs include unique loops in blue here. In fact, these loops were noted way back in January, right after the SARS-CoV-2 sequence was reported, and these loops kind of set SARS-CoV-2 apart from the other sarbeco coronaviruses. So what Enya did is he replaced these loops with the corresponding sequences in closely related SARS-CoV-1, the 2003 SARS, and he called the resultant replacements delta NTD for, changed loop or changed NTD. And so he evaluated the consequences. As you can see here, Enya found that the NTDs, they must display the SARS-2 loops to suppress the cell fusion reaction that he uses. This came from these additional cell fusion assays, this time comparing the NTD structures with the delta NTD loop, both very pure, both at 10 micromolar concentrations to impede the fusion. And so he's done this many times. And at the right here, only the NTD suppresses fusion, the delta NTD loop does not. So either the loops are responsible directly or indirectly for attachment factor binding. But what both Enya and I found more interesting, though, is that these changes in the NTDs, they also altered the later stages in the virus entry process. So I take you through the later stages of the virus entry process at the left, and you can see in the Western blot data at the right that the delta NTD viruses have a particular propensity to shed their S1 from the transmembrane S2. This was done with virus-like particles, and it really, using these SARS-CoV-2 virus-like particles, they actually found that this propensity to shed S1, it just focused on the middle lane of the Western blot, was particularly enhanced in the delta NTD viruses. Membrane-bound S2 fragments are uniformly similar across all tested VLPs, but the delta NTD virus-like particles were set apart by having very little S1 caps on their spike. So somehow, the NTD loops, even though they're very distal to the S1, S2 binding interface, they're central to both binding some as yet unknown attachment factors and to controlling the stability of the S1, S2 heteromers. So there's more, there's a little more that he found, and I can take you through this. And you also found that changes in the NTDs altered the sensitivity of viruses to proteolytic activation. So finding this out required a new assay, so I'll go through this slide somewhat methodically here. At the top left, once again, part of the diagram depicting the execution of virus entry and membrane fusion, now with the activating proteases, oftentimes transmembrane proteases operating as fusion triggers. And what we found out relatively recently is that we can replicate this process of virus cell binding and fusion fairly easily using simple in vitro reagents. What we use are virus-like particles that have nucleocapsids that we have tagged with a nanoluciferase fragment. So that's depicted here at the bottom left. And we purify these virus particles and we mix them with purified, also highly purified extracellular vesicles that we have engineered to harbor ACE2, primary protein receptor for SARS, of course, that are linked then to intraluminal complementary nanoluciferase fragments. And so all it really takes is a mixing of these two particles in the presence of an activating protease. We use trypsin at this time, and voila, boom, there's membrane fusion between the vesicles and a strong complementation into easily measured nanoluciferase. So the levels of active nanoluciferase, therefore, are the readouts for coronavirus membrane fusion. And at the right's an interesting finding that Enya obtained. The delta NTD loop viruses, as VLPs, are sort of hair triggered for membrane fusion. It takes extraordinarily low levels of trypsin to activate these delta NTD viruses for vesicle fusion, while native SARS-CoV-2 VLPs, either the earlier, D614 form of the SARS-2 or the now more prevalent variant isolate D614G, they require a much more trypsin, kind of more of a trypsin push, if you will, to become activated for the fusion reaction. So those are the kinds of data he's finding. And so as it says here on this slide, the SARS-2 NTD loops, distal from the S1, S2 interface, they contribute to cell binding to as yet unknown attachment factors, and they also have roles in regulating post-binding entry stages. Just 2 points here. I mean, we could understand this much better with more structural data. Structures will bring more answers to questions about receptor-induced changes and interdomain communications between the spike proteins. There's already several papers on this topic, so we're learning quickly from others, mostly structural biologists, about the, how do you say, the gymnastics of these spikes. And that might help us to get a sense of how changes distal act, how do you say, allosterically to induce fusion-ready states. And second point that I wanted to make is that the NTD loop changes that Enya made, they're, of course, they're artificial, but I think his findings do say something about the selective pressures on viruses as they adapt. You change the NTD structures, and it does seem possible to obtain a virus that's particularly fusion-ready, primed to enter a cell that has very, very little activating protease as a susceptibility host factor. So this would seem proviral. As infection, of course, then it'd be less dependent on host proteases, and the viruses might be able to expand to new niches. But it's important to note, of course, that such an adaptation comes at a cost, and the cost, at least in this instance, is the high cost of spike protein instability. And so these are the sorts of selective forces that I think might be central to restricting adaptive changes in the spike proteins. And I think we should watch out for those kinds of changes as coronaviruses evolve zoonotically. So I want to finish just with a few slides to promote this in vitro virus-like particle extracellular vesicle fusion assay I told you about. Here the assay is depicted again with additional stages, binding of the two vesicles, spike binding to ACE2, hypothetical tethering of the two vesicles via extended S2 intermediates. This may be, in fact, in the middle, fairly durable intermediate state. Because we have some evidence for that. And culmination of fusion via proteolytic activation. So it's not lost on us that there may be several uses for this assay because it reflects authentic SARS-CoV-2 entry. It's cell-free, BSL-1, uses durable reagents. It's easy to perform. It's amenable to high throughput screens. We use multi-well plates, of course, and we get Z factors of around 0.9 for this assay. So there's really excellent signal to background in these assays. And so the assay can measure neutralizing.
You got two minutes.
So we can measure neutralizing antibodies very, very readily. We've communicated, we have, at the left here, you can see IC50 neutralization values for negative, intermediate, and high titer patient sera. And I just want to point out that the assay has really excellent concordance with BSL-3 plaque reduction neutralization titers, PRNT. So we get data, as you can see in the blue table here, very close with, very similar to BSL-3 assays. So we think this assay is a good adjunct to the ELISA tests that use pre-fusion stabilized spike ectodomains as antigens. And in fact, Susan Baker and Susan Uperchurd and I are doing comparative analyses of the ELISA and the neutralization assays here. There's many domain-specific neutralizing monoclonals that could be compared for relative potency because this assay measures obviously more than the common RBD-specific neutralizers. And then the assay can measure fusion inhibitors. We've communicated and collaborated with Matteo Parado and Ann Moscona, who've developed several peptide-based inhibitors of SARS-CoV-2 in red here. And we've evaluated them with this in vitro assay. And the peptide we titrated here is one that blocks this fusion cascade at 30 nanomolar IC50, had a measured IC50 of 17 nanomolar in a cell-based assay, so good concordance there. And finally, the last slide, I just wanted to say with this, we just got this assay going, but we have more to consider. We can do many things. We can evaluate different activating proteases with the assay, fusion kinetics. We can set up temperature, pH, and ionic strength conditions similar to that in the endosome where the unleashing of the virus fusion may occur. And we have ways to incorporate attachment factors, different ones, into the vesicles that we use as surrogates for cells. And so we can perform the assays in that way too. So there's a lot more to learn. We'll keep learning safely. Anya, this is Anya Ching here, learning safely. And we work with Binod, Grant, Tom, and Emily operating every day, masked, of course. And so thanks for listening. That's my story.
Thank you, Tom, for a really beautiful presentation. Now open for questions.
I could ask one. Hi, Tom.
Yeah.
Beautiful.
Yeah, sure.
I agree. All the fusion assays are so important, and they're not done yet enough. So this will be really handy. So I wondered, had you yet looked at other proteases in the assay than trypsin? I mean, it seemed that you need really huge amounts of trypsin, which I'm sure is not the right guy in reality. So I wondered if the authentic proteases were much more potent.
Yeah, good question. Yeah, we've tried to the TMPRSS2, also a serine protease like trypsin, actually structurally very similar to trypsin. But we have some challenges incorporating the TMPRSS2 into these vesicles that we use as cell surrogates because the TMPRSS2 in fact has ACE2 as a substrate. So TMPRSS2 expression cleaves the ACE2 ectodomain. So that causes some issues, but I think we're working on getting soluble forms of TMPRSS2. The other are cathepsins, which we've got in the pipeline. So, yeah.
Right. Jahar, you have a question?
Right. Thanks. Tom, that was a terrific presentation. Thank you very much. We are trying to understand how SARS-CoV-2 spike protein fuses with lung cells in situ in a mouse model that expresses human ACE2. But The question I have for you is, and so we're very excited with the fusion model. The question I have is, do you anticipate differences between the SARS-CoV-1 spike protein and the CoV-2 spike protein in the way they fuse with host cells, and why there should be a difference? Because they seem to have different propensities to fuse with different types of cells in the lung.
Yeah, I think you could expect a big difference between SARS-CoV-1 and SARS-CoV-2 that primarily traces to that priming cleavage that I was referring to near the beginning of the talk. The SARS-CoV-1 spikes are not susceptible to priming cleavage. So uncleaved spikes on SARS-CoV-1, how can I put it? They need more proteolytic activation over on the target cell side. And that's usually achieved only after the viruses are endocytosed into a really cathepsin-rich environment, and cathepsins chew away at SARS-CoV-1 spikes and activate the fusion. So the time and place of SARS-CoV-1 fusion wouldn't be the same as SARS-CoV-2, because SARS-CoV-2 with its cleaved and primed spikes is a little more sensitive to a immediate cell surface proteolytic activation by serine proteases. Okay, so I think those are the key differences between SARS-1 and 2. There's other differences as well. I mean, SARS-2 has higher affinity for ACE2 than SARS-1, but
Thank you.
I think it mostly boils down to the priming cleavage. Yeah.